So I would like to start with an introduction on the background rates of AESI for COVID-19, and I am Miriam Sturkenboom and scientific coordination of the SPEAC team, and SPEAC is a project and has an executive board, and I'm talking on behalf of them. And the project is led by Dr. Robert Chen, Corey Decker, Steve Black, Andy Stargagis, Wanting Wang, Flo Munoz, Margaret Gerwith, Dale Nordenberg, Barbara Law, and Angel Honrado. Next slide, please. So just a brief introduction about SPEAC itself. SPEAC stands for Safety Platform for Emergency Vaccines. It's a project funded by the Coalition of Epidemic Preparedness and Innovation, CEPI, and it provides a vaccine safety monitoring service for CEPI, starting with clinical trials. Why do we do that? What we know is that vaccine safety monitoring is of crucial importance for maintenance of trust in vaccines, and to monitor them adequately, it's really important to have tools, guidance, templates available so that we can actually monitor it in a harmonized fashion. And this is particularly important if CEPI goes to their 100-day aspiration. Next slide. So what does SPEAC provide? It does a lot of reviews and synthesizing existing literature in a systematic to make list of adverse events that need harmonized monitoring, their risk factors and background rates, case definitions, and data collection forms. SPEAC also reviews clinical trial protocols for vaccine developers that are supported by CEPI. It provides continuous safety monitoring support to CEPI. SPEAC trains experts for DSMB members in LMIC through an online course that they have created. SPEAC synthesizes available knowledge on the vaccines in vaccine benefit risk templates, and it prepares for active surveillance monitoring of vaccines in LMIC, and it disseminates tools and information and measures the impact. Next slide. For COVID-19, SPEAC supported CEPI by reviewing the literature, and it created a list of adverse events of special interest, which was updated periodically and made publicly available. And this list has been utilized widely. We will see that later. SPEAC also created companion guides of the AESI, systematic reviews of rates and risk factors, and created new case definitions where they were needed. This list and the materials have been utilized widely. Several research networks were requested by regulatory authorities and CDC to generate background rates just prior to the vaccine rollout. And this was the Access project that was funded by EMA and ran by the research networks Back4You and the EUP and PV network. The BEST initiative funded by FDA that was conducted by the Odyssey network, and the GCOV study that is funded by CDC and conducted by the Global Vaccine Data Network. Next slide. Thank you. So here you see that the publication that was written by a core group of the executive board from SPEAC, the critical role of backgrounds of possible adverse events. And here in this article, you can also find the list that was prepared by the SPEAC project, and which one has been utilized by the various regulatory agencies. I'm not going to go through all of these events, but this was the basis for the monitoring. Next slide. In this article, we also describe what are the key factors to be considered when using background rates. And first of it is that many background rates that are produced are crude rates, meaning that the events have not been validated. They are often based on diagnosis codes. And this should be considered when comparisons are done. We also noted and showed that incidence rates of AESI may vary by multiple factors. And this is, for example, gender, age, calendar time, the method for AESI measurement, healthcare utilization patterns, which may change over time, geography, and the population studied. Importantly, that all these factors should actually be considered when observed expected analyses are conducted. Next slide, please. As a preparation for the rollout of the COVID-19 vaccines, several networks conducted and created these background rates. And here you see an overview of the ones that were published and made available in February 2021. There was the first release of the Access Project background rates in countries in the EU. Then there was a publication in June by the AESI network that also had rates from the US and Japan. In June 2021, Gubernor also published a literature review of US-based background rates. In 2022, there was a release of the GVDN background rate dashboard with 13 AESI from 8 countries. And there, in between, there was also the release periodically of the SPEAC companion guides for selected AESI that also contained a systematic review of background rates globally. Next slide. If we look at these, some of the results, what we see here is some results from the Access project, and here used for only 8 of the AESI. And what is important here is that these line graphs actually on the x-axis you have age, so you can see that the incidence rates change by age, some of them very much actually, but you also see different colors, and the colors actually indicate the different type of way that diagnostic codes can be obtained, and we call that provenance of the events. And for some of them, some data sources only capture in-hospital data, others capture inpatient and emergency room, others capture only GP. And what we show here in this paper is that actually the rates vary quite a lot by provenance of diagnostic codes. So also this is a factor that needs to be considered when observed expected analyses are conducted. Next slide. This is an example of the AESI study that comprised both countries in the EU, but also US and Japan. And this is their publication, and you can see the graphics, and one can already note that on the y-axis, the range is quite large. For some of the events, the rates change 100-fold between one or the other data source. And you see also the age and gender patterns. So in this instance, we would say pooling needs to be done carefully across data sources because there is considerable heterogeneity. And this is the piece of the table that was published where age-specific and gender-specific rates were done, but they were pooled across all of the data sources. Next slide. So for the GVDN website, rates take age, gender, and provenance of diagnostic codes into consideration. And if one wants to utilize the rates, there is a nice dashboard by country and by data source. And the rates can also be downloaded in an Excel sheet. Next slide. So the purpose of this webinar was that a lot of the participants and a lot of researchers have been working to generate these background rates rapidly prior to the rollout of the COVID-19 vaccines. And now that times are a bit quieter, it would be good to look back and see like what worked and what did not, and how should we be better prepared the next time. And therefore, we are very grateful to our colleagues from the regulatory agencies and CDC to participate in this workshop, and all the people that have signed up to listen. And hopefully, if you have questions, please feel free to put them in the chat. Thank you very much. Then I would like to go to the next series of speakers, and I would like to announce our colleagues from FDA, Dr. Narayan Nair, who is the director of the Division of Pharmacovigilance in the Center of Biological Evaluation and Research and the US FDA. And he oversees the division that performs pharmacovigilance activities for vaccines and other biological products regulated by FDA. We also have Dr. Brendan Day, who is a pharmacovigilance reviewer and team lead in the Division of Pharmacovigilance of the Office of Biostatistics and Pharmacovigilance in CBER. Thank you very much for being present here today and sharing your experiences and lessons learned with us.
Hey, good morning. This is Brendan.
Just checking if
Narayan is
on. He had some
technical
technical issues
earlier.
I can see Dr. Narayan there, I think.
I think that Brendan Day is the person that's talking now. We can hear you, but we hear you with a lot of echo. Could you please try again?
Okay, is that better?
Yes, much better.
Okay, thank you. I think Narayan might not be able to unmute for some reason, so I'll just go ahead and get started. So again, I'm Brendan Day. I'm a pharmacovigilance reviewer in CBER at FDA, and I'll just preface this by saying that we're going to be talking about our division's work, and we focus on passive surveillance. So this is going to be FDA's approach to use of background rates and observed to expected analyses. And although our CDC colleagues aren't giving a separate presentation today, we do work very closely with them for evaluation of potential safety signals. And so I'm going to be presenting some analyses which we collaborated on. Next slide, please. So first, a disclaimer. My comments are an informal communication and represent my own best judgment. These comments do not bind or obligate FDA, and mention of a product or company name is for identification purposes only and does not constitute endorsement by FDA. Next slide, please. So the Vaccine Adverse Event Reporting System, or VAERS, had been receiving a steady number of increasing reports since its inception in 1990. And with the advent of the first COVID-19 vaccines and the first EUA in the United States in December 2020, we experienced an unprecedented surge in adverse event reports. And it's within this context that in our division, we had to adapt our surveillance strategies to meet the needs of this large increase in adverse event reporting. And one of the tools that we found particularly helpful at this time was the use of background rates and observed to expected analyses. And so in this presentation, I'm going to provide 3 examples where we used observed to expected analyses to further evaluate potential safety signals and ultimately assist in regulatory decision-making. Next slide, please. So first, I wanted to put reporting rate comparisons and background rates within the larger context of vaccine safety surveillance at FDA. So in our division, we perform signal detection and signal evaluation activities. And for the routine activities for signal detection, we have individual and aggregate ICSR, or individual case safety reports. We perform data mining for VAERS reports. We regularly review the literature. We review sponsor-submitted periodic safety reports, and we generate our own internal surveillance reports. Of course, we can identify safety signals from other areas, such as safety data from ongoing clinical trials, media reports, or communications with other health authorities. Once a signal is detected, we have a number of methods available to us for further evaluation. And these are just some examples. And the methods chosen will depend on the safety signal being evaluated and the product in question. But the methods available could include an ICSR review, generation of a case series, performing a reporting rate comparison to background rates, pharmacoepidemiology study, another look at clinical trial safety data, or asking the sponsor to perform their own assessment of the safety signal. Next slide. And so it is within this context that we see reporting rate comparisons are just one tool within this within our signal evaluation methods available to us. Next slide. At this point, I should point out that although reporting rate comparisons to background rates are something we were able to do before the pandemic, one thing that changed with the pandemic was our access to high quality and timely vaccine administration data. So this slide shows the CDC's COVID data tracker, and we worked closely with CDC to obtain vaccine administration data for a lot of these observed to expected analyses, which ultimately made these analyses more accessible, and we were able to perform them for a lot of the potential safety signals. Next slide. So now the three examples. First is myocarditis and the mRNA COVID-19 vaccine. Next, thrombocytopenia and the Janssen COVID-19 vaccine. And third, Guillain-Barré syndrome and the Janssen COVID-19 vaccine. Each of these analyses are now published articles, and I include the references at the end of this presentation. Next slide, please. So first, myocarditis and the mRNA COVID-19 vaccine. For this analysis, this is a CDC-led article, and for this analysis, CDC and FDA authors obtained a number of VAERS reports of myocarditis after either the Pfizer-BioNTech or the Moderna COVID-19 vaccine in individuals aged 12 years and older. And for a background rate, they used IBM market scan data, which is claims data from 2017 to 2019 to estimate age stratified rates of myocarditis. And they applied a risk window vaccine type and dose number. Next slide. This slide shows the main analysis or the main results for this analysis. And on the top half of this table, we have males, and on the bottom half, we have females. And across the table, we have the Pfizer-BioNTech and the Moderna vaccines stratified by dose. And on the far right column, we have the expected cases of myocarditis for each of the age groups. And what we can see from this analysis is that for many of the age and sex subgroups, we have elevated observed to expected counts. And this is most prominent for adolescent males and young adult males, particularly after the second dose. And although females had lower rates in general than males, they also had elevated rates for these same age groups. So this analysis ultimately contributed to labeling updates for the mRNA COVID-19 vaccine. Next slide, please. For the second example, we have thrombocytopenia and the Janssen COVID-19 vaccine. In this case, this safety signal was identified after the TTS safety signal, or thrombosis with thrombocytopenia syndrome signal. So in this case, reviewers were concerned about a separate safety signal just for thrombocytopenia. And in this case, they reviewed VAERS reports for thrombocytopenia, where there was a laboratory value reported less than 150,000 platelets per microliter, or simply a diagnosis of thrombocytopenia. And they compared these rates to an expected rate of 3.3 per 100,000 person years from the published literature. They applied risk windows of 7, 14, 21, and 28 days. And they also performed sensitivity analyses, varying their case definitions, making it more stringent or less stringent. Next slide, please. This slide shows the main results for their analysis. Down the left-hand side of this table, we have the risk windows of 7, 14, 21, and 28 days. And down the right-hand side, we have the rate ratios. And from what we can see in this analysis is that all of the rate ratios are elevated, with the lower bound of the 95% confidence interval around 2 or greater, suggesting that the observed number of cases of thrombocytopenia are at least double what we would expect based on the published background rate. And so this analysis ultimately contributed to the regulatory decision to update the labeling for immune thrombocytopenia. Next slide, please. And for our last example, we have Guillain-Barré syndrome and the Janssen COVID-19 vaccine. In this case, reviewers were concerned about increasing reports of GBS after the Janssen COVID-19 vaccine. And so they gathered cases for this observed to expected analysis. I should note that at this time, they were concerned that there wouldn't be enough medical records to perform a rigorous application of case definition. And so they defined cases as presumptive cases of GBS with some clinical criteria and measure preferred terms but they did not apply a Fulbright and collaboration case definition. And for the expected rates, they used age stratified rates provided by a meta-analysis by Sedgbar and colleagues. And they applied risk windows of 21 and 42 days. And finally, for a sensitivity analysis, since they had not received all the records for cases, at that point, they assumed that when records did come in, 80% would meet Brighton collaboration case definition. Next slide. And this slide shows the main results for this analysis. On the top half of this table, we have the time onset of within 21 days, and on the bottom half, we have the risk window within 42 days. And each of these analyses are age stratified. Across the table, you can see the input data for the observed to expected analyses. And on the far right column, you have the rate ratios. And what we can see is that all of the rate ratios are elevated, and the vast majority of them are significantly elevated. And so this analysis contributed to the regulatory decision to update the labeling for Guillain-Barré syndrome. Next slide, please. So in conclusion, although reporting rate comparisons have been a part of FDA's vaccine safety surveillance before the pandemic, the volume and intensity of the vaccine surveillance work increased, and we also had better access to vaccine administration data. Ended up playing a critical role in the timely evaluation of several safety signals for COVID-19 vaccine. Next slide. And thank you for your attention.
Thank you very much. Let's continue with the next presentations. We do have some questions in the chat, but we will take them at the end. Pia,
Next presentation is from EMA, yes,
okay,
and they are going to share the screen. Perfect.
Okay, good. So let me introduce then our colleagues from EMA. We have Catherine Cohet, who is a senior pharmacoepidemiologist in the real-world evidence workstream of the Data and Analytics Methods Task Force of the European Medicines Agency, and she focuses on international guidance on real-world evidence and on public health crisis preparedness, a steering group member of the joint EMA-ECDC vaccine monitoring platform. We also have Maria Gordillo, who is the data science specialist in the healthcare in the same group at EMA, and she focuses on pharmacovigilance activities related to the COVID-19 pandemic and analysis of real-world data to support regulatory decision-making. Thank you for joining us, and please, if you can manipulate, or you can move the slides yourself, yeah.
So many thanks for the introduction, and good morning, afternoon, or evening to all the attendees of this webinar. I would like to thank the organizers for the opportunity to present today. So as Miriam introduced, I am Maria Gordillo Marañón, and I work as a data scientist in the healthcare data workstream at the Data Analytics and Methods Task Force in the European Medicines Agency. And I'm here today with my colleague, Catherine Cohet, from the real-world evidence workstream. So today we will present on how observed to expected analyses were used for the safety monitoring of COVID-19 vaccines at EMA, including 2 case studies, thrombosis with thrombocytopenia syndrome and myocarditis. And we will end the presentation with the lessons learned during these past 3 years. So I'd like to begin by providing some context about the COVID-19 vaccination in the European Union. So since the vaccines were approved, they have been extensively used in the EU, with over 975 million doses administered as of the 20th of the current month. This high vaccine uptake led to an unprecedented volume of safety data from spontaneous reports, with over 1.7 million cases of adverse drug reactions originated in the European Economic Area, and reported to Eurovigilance, which is the system to collect, manage, and analyze suspected adverse drug reactions to medicines authorized in the EEA. So for the EMA, the nearly 2 million reports of adverse drug reactions received pose a challenge for pharmacovigilance activities. We can see in this graph of the number of cases received per month that the number of reports related to COVID-19 vaccines increased exponentially as soon as the vaccines were deployed in December 2020. Due to the nature of the COVID-19 pandemic and the uptake of the vaccines, the reporting of suspected adverse reactions had been anticipated to be very high, much higher than for the 2009 H1N1 pandemic. And indeed, this prediction was confirmed because within 3 months of the start of the deployment of the COVID-19 vaccines in the EEA, the total number of reports for these vaccines in eurovigilance was almost 20 times that of H1N1 pandemic vaccines in the same time frame. Also, whereas reporting levels for H1N1 vaccines reached a plateau after a few months, for the COVID-19 ones, they have continued to grow steadily. Also, compared to the H1N1 vaccines, the COVID-19 ones, they were not pre-pandemic or mock-up vaccines, and this situation led to a fast development of vaccines which were authorized following a rolling review, which is a regulatory tool used by the European Medicines Agency to speed up the assessment of promising medicines during public health emergency without compromising quality, efficacy, and safety. So this whole experience led to numerous lessons learned on methods, data sources, and collaborative work. And we will see some of this in the next slides. So here we can see a timeline of the authorization dates for the COVID-19 vaccines in the EU, as well as the specific activities put in place by EMA to ensure the enhanced monitoring of emerging vaccine safety information. And this included preparedness activities before the rollout of the vaccine, such as the launch of the Access project, which estimated background incidence rates of adverse events of special interest, and also generated template protocols for safety of COVID-19 vaccines. And also those activities that focus on early and continued safety monitoring and safety and effectiveness studies following vaccination of the population. So this set of pharmacovigilance activities included ad hoc approaches to generate evidence for signal evaluation. And one of these methods, which was introduced at the beginning of the presentation, are the observed to expected analyses, which compare the number of observed cases of an event following vaccination to the number of cases that would have naturally occurred in the same population without vaccination, which are called expected cases and are estimated from background incidence rates. So to perform an OE analysis, one needs 3 key variables: the observed number of cases, the risk period or the time period where there is a suspicion or biological plausibility that there is a vaccine-associated increased risk of experiencing the event, and the expected number of cases. So in OE analysis conducted by the EMA, the observed cases are based on spontaneous reports in eurovigilance from the EEA. The risk periods are generally determined based on published information, biological plausibility, or time to onset distributions. And as for the expected cases, these are calculated using COVID-19 vaccine exposure data that is sourced from the European Center for Disease Prevention and Control website, together with background incidence rates generated by the Access project or ascertained from healthcare databases to which EMA has access to. In addition to the main OE analysis, there are also sensitivity analysis performed with different risk periods, background incidence rates, or case definitions. So in the next slide, I would present 2 safety signals in which the OE analysis were instrumental in characterizing the risk. So on the 7th of March of 2021, the Austrian Agency for Health and Food Safety alerted the EU regulatory network to unusual cases of thromboembolic events following administration of Vaxzevria. A few days later, the EMA's Pharmacovigilance Risk Assessment Committee initiated an evaluation of thromboembolic events, which led to the identification of a new risk for a rare combination of thrombosis with thrombocytopenia. And the evaluation of this signal was challenging because when the signal emerged, there were no case definition, background incidence rates, or specific metro attempts. So the early OE analyses were based on proxy background rates that including coagulation disorders, disseminated intravascular coagulation, and cerebral venous sinus thrombosis or CVST. So here we can see the results for CVST. So as we can see on the forest plot on the right, the number of observed cases following the administration of Vaxzevria was higher than expected, especially in the younger age groups. And this is highlighted with the red bars in the plot. So this analysis contributed to the assessment report of the signal, and by the 24th of March of 2021, the product information of Vaxzevria was updated and healthcare professionals and the public were warned about the risk. The next case study concerns myocarditis. And for this event, the starting point was different because myocarditis and pericarditis had been reported in association with SARS-CoV-2 infection and were therefore included in the list of adverse events of special interest. So as you probably remember, in early 2021, a signal of myocarditis emerged in Israel for Comirnaty, where most cases occur predominantly in young male vaccinees after the second dose. So this triggered a review of myocarditis and pericarditis for both mRNA vaccines at EU level. And as of May 2021, 145 cases of myocarditis had been reported to Eurovigilance from EA countries for Comirnaty. And therefore, Prague requested an OE analysis of myocarditis and myopericarditis, which was performed according to the input data shown in the slide. So here we see the results of the analysis stratified by age, gender, and vaccines, where the results at the top correspond to Comirnaty and at the bottom to Spikevax. So the statistically significant OE ratios over 1 are highlighted in red, which allows to rapidly identify the groups at risk, which in this case were males between 18 and 24-year-olds. So the occurrence of cases of myocarditis and pericarditis was reflected in the product information of these two vaccines and communicated to healthcare professionals and the public. So from the implementation and interpretation of OE analysis, we have learned very valuable lessons on facing a massive influx of spontaneous reports, on comparability between observed and expected cases, on dealing with incomplete data, and on generating, selecting, and using background incidence rates. So let me give you some specific lessons in each group. First, we have learned that OE analysis provide an additional tool for safety monitoring of COVID-19 vaccines, which provides a one-to-one comparison and is therefore independent across vaccines. And this is not the case for other statistical tools, such as the reportings or ratio, which takes into account all the medicinal products in the spontaneous reporting database. However, we have noted that one of the main limitations of OE analysis is the underestimation of observed cases that may occur due to underreporting of some events or delayed reporting. Second, we have learned that a poor alignment between the codes used to calculate the expected cases and to identify the observed cases may affect the robustness and interpretability of the OE results. And third, with missing and incomplete data is a challenge, and we had to make assumptions and imputations to deal with it. Lastly, there are lessons learned on generating, selecting, and using background incidence rates, and I will hand over to my colleague Catherine to present this.
Thank you very much, Maria. As shown by Miriam and Brendan earlier in this webinar, there were a number of sets of background rates available throughout different organizations and different consortia in early times of the pandemic. So this highlights the first lesson, which is about preparedness, and Miriam has touched upon this in her introduction as well. So we had background rates for cardiac events, such as myocarditis, but we also needed to be prepared for the unexpected and able to respond quickly and leverage available data generation frameworks and collaboration, as was the case with TTS. We have also learned that as prepared as we could be in terms of looking at the right event, sometimes some additional or different level of stratification was required. The same remark in terms of availability and preparedness for case definitions or case finding algorithm, phenotypes, etc. So TTS, again, is a good example here. In terms of representativeness, it might have been challenged at national level when countries updated their vaccination recommendations. However, this could be considered more as a theoretical issue for Europe, where large differences between countries would not necessarily be expected. And finally, the hot topic, heterogeneity between data sources. This is actually not really a new topic, but it has received quite some attention lately with the increasing use of multi-database studies, and it received particular scrutiny in the case of COVID-19 vaccine safety evaluation. However, heterogeneity can be informative. It could, for example, reflect true population differences between data sources. And there are a number of tools to address it. This will certainly be the topic of another full webinar, so I will not expand here, but I'm happy to take questions. Just to mention that leveraging metadata catalogs, reflecting at study design stage when generating background rates, and using meta-analytical methods can all help addressing heterogeneity. I indicated on the slide the title of an EMA manuscript on the topic that is under production at PDS, Pharmacoepidemiology and Drug Safety Journal. It should be available online very soon, probably it's a question of days. And finally, when it comes to public communication and risk communication, visualization tools such as forest plots should probably be systematically used. So in conclusion, a few key messages. I think we all agree that the pandemic has been a huge booster, not only for the development of tools and methods to deal with large volumes of safety data, but also to improve evidence generation based on secondary use of data. From a regulatory perspective, the need for flexibility and efficiency and the value of scientific collaboration have been important lessons learned. Miriam asked in our introduction what we can do to maintain preparedness, and one endeavor would be to reflect on how we can improve and update all those available repositories of background rates. And there are many usual suspects for which adverse events of special interest we should maintain background rates for, and GBS is an example as shown in the FDA presentation just before. Finally, I believe we should continue to reflect on how we can still today complement lessons learned also from H1N1, and how we can apply learnings from H1N1 and from COVID-19 to any types of vaccines. And with that, I thank you for your attention, and I guess it's question time. Thank you.
Well, thank you very much, Catherine, and thank you for the very good explanations and for the presentations. There are a couple of questions in the Q&A. So for people, if you have a question, please post it in the Q&A. I would like to start with a question from Trezor Bojic, who has actually multiple questions in the Q&A. I think the first one I would like to address, or have to be addressed by the people, is how often ideally do we need to update background rates? So is there anybody who would like to give an answer to that? Catherine, maybe because you just asked for that. Would you like to?
Yes, well, how often? I guess it's the key question, and maybe here we could differentiate the type of adverse events, which I think I use the term usual suspect. We know, for instance, Guillain-Barré, multiple sclerosis, all those series of immunological or neurological conditions that we know are background rates of interest for any types of vaccines. So we should probably have a data bank or repositories for those adverse events of special interest. And now, of course, there's always those such as TTS, such as narcolepsy at the time of H1N1, where we will not be able to predict. But I think we could probably easily come up with a solid list of background rates to be updated on a regular basis.
Thank you. Then I would like to continue with a question from Dr. Walter Strauss from Moderna. Can you speak to challenges and possible solutions for establishing background rates where the events are exceedingly rare, such as, for example, capillary leak syndrome, or where case definitions are challenging? Is there anybody who would like to speak to this? Colleagues from FDA, EMA? No, nobody.
Miriam, it's Walter. Perhaps I could
Yes.
just provide a bit of clarification.
Yeah.
The challenge for this one is that there were fewer than 500 reports of capillary leak syndrome prior to, I think, 2017. So the notion of background rates is really, really challenging. So let me turn it over. That's the background to this.
Okay, so I don't hear anybody wanting to respond. Is there somebody who would like to answer this question? Otherwise, I can try. I think the capillary leaks for some of the events that occurred, like our new syndromes, or for example, also with MIS, it was the case, and from capillary leak syndrome, the problem was that there were no codes really assigned to it in the regular, diagnostic vocabularies. And that is a problem then in generating the background rates if you use these electronic health record data sources. So then you would need to rely on either literature or primary data collection or other ways of data collection. I think what we are seeing currently is that the availability of those rates they can be made rapidly available if you can identify the event in an electronic healthcare data source. So I don't have an answer to this question in terms of how we should deal with it. There are other questions in the chat also about background rates for example, Sub-Saharan Africa, also from Trezor Bocic. And also here we are seeing that oftentimes these rates are not available. Maybe Barbara, do you want to say anything? Barbara Law, who conducted a lot of the systematic reviews to retrieve the background rates.
Yeah. Hi, Miriam. Sorry, no, I don't think I have anything to add.
Okay, so that is the limitation we currently. Is there anybody else who would like to answer this? Please raise your hands then.
Miriam, it's Catherine again. Maybe just a few more thoughts. And I was going through the questions, and I see there was also a question from Rebecca Chandler about doing actual so-called signal evaluation studies. So I guess this is like more formal pharmacoepidemiological or inference studies. And I think we also, and I totally agree with what you said, it's very difficult when it's a very rare event, when it's an event we have not planned for. But I think we should also contextualize the use of observed to expected analyses more in a continuum of vaccine safety evaluation. This is really the first step when we have a signal emerging. We are just trying to act as fast as possible using the best data available, but it does not replace an actual more robust evaluation. So just recontextualizing this in a longer term evaluation, also using actual pharmacoepidemiological study designs.
Rebecca, does that answer your question? Thank you, Catherine. Or would you like to?
No, I just was going to make an observation that it does seem, though, that in this particular instance, I mean, I believe I noted both from the US and the European presentations that labeling changes were done based on the observed versus expected analyses examples.
Yes, this is
This is Brendan. Sorry, go ahead.
Oh, yeah. Can people hear me? Am I off mute?
Yes, we can hear you perfectly.
Okay, yeah. So sorry, I had some technical issues and was not able to get off mute earlier. But this is Narayan Nair with Division of Pharmacovigilance, and just to comment that is correct. I mean, we typically think of the passive surveillance as hypothesis generating, our early warning system in the US, but we did have instances where it was a very dynamic situation. And we did do labeling changes based on the OE, based primarily on passive surveillance data. Now, oftentimes we had active surveillance data that informed that, but you know, the deciding factor was information from the passive surveillance. And one of the reasons we were able to do that with the COVID-19 pandemic was we had very good denominator data, since this was a federally mass vaccination clinic, and the vaccinations were distributed by the federal government. We were able to gather information, our colleagues from CDC, on doses administered, and it was quite granular in terms of the age distribution and how many individuals had received a dose. And that did allow us to do much more robust analyses than we typically have been able to do with other vaccinations in our passive surveillance database.
So Miriam, it's Bob Chen, if I may. So one challenge, obviously, with passive surveillance is the point that was raised by Maria earlier in terms of underreporting. And as I assume that you all in your signal generation did not build in a factor to account for the underreporting for each of the different AEs. And so one suggestion going forward is that in the case of intussusception after rotoshield, Tom Verstraeten was able to do a capture-recapture study that showed that the completeness reporting for intussusception was basically about half, 47% completeness to VAERS, despite the publicity. And one would say that if your kind of threshold for generating that signal did not take that into account, it may potentially double the time before an alert actually is generated. So it would be important for us to go back retrospectively to do as many capture-recapture studies as possible in different settings and countries, wherever that's possible, for different outcomes to better understand that degree of underreporting and to better calibrate that kind of whatever the alert signal that we want to use going forward. Over.
Thank you, Bob. There are many more questions now in the Q&A. I would like to ask Agnes Kant. You have several questions. Would you like to explain them live? I don't hear her. Okay, so then there is another question. There are questions related to correction for underreporting, whether any of the agencies has done that in their observed cases.
I think Agnes is trying to speak, Miriam.
Okay.
Yes,
Agnes, okay.
yes, sorry, I was muted. Yes, I had two questions. The last question is what you ask is, has someone experience correction for underreporting, like Bob just mentioned, because we don't know how to do this. So if there's experience in this kind of analysis, I would probably want to know how people did this. And the other question is, if strata are a little bit comparable outcomes per stratum, did you also try to make a sort of standardized observed expected ratio? Because we did that in some cases of our analysis, and then you have a more narrow confidence interval. So two different questions.
Thank you, Agnes. Is maybe first our FDA colleagues, did you do any correction for the potential underreporting?
This is Narayan,
Sure,
and Brendan, please chime in. We did not do any correction for underreporting. I think it's a very good and important point. We did have mandatory reporting requirements for healthcare providers and for vaccine manufacturers, regardless of attribution. We had had this reporting requirement that individuals report serious adverse events and various others that we listed. But nonetheless, we still, there certainly probably was underreporting, as well as stimulated reporting, which we would see if an adverse event would appear in the media. So we did not do any correction, but it's something that we were very conscious of as we were looking at these numbers in a qualitative sense.
Mhm.
Thank you very much. Any EMA from?
Yes, Maria here. So we did look into underreporting through the sensitivity analysis. So it's very difficult to estimate the level of underreporting for each of the events, but we look into different scenarios with different levels of underreporting and see the effect that would have in the estimates. So it's something that we include in our approach.
Okay, thank you.
Thank you. Okay, then I would like to continue with a question from Jim Guthrie. Jim, do you want to explain it yourself? Otherwise, I can read it. Oh, you're there. Yes, go ahead.
Thanks. Yeah, I'm wondering if you can comment on the complexity where the wild type infection during the pandemic may impact the expected rates of adverse events of special interest, given that most of the background rate data available usually at the time is pre-pandemic, before circulation.
Anybody would like to address that? I think that's, for example, for all the cardiac events.
Yes, Miriam, maybe I can take this. What we were doing, we were looking both at background rates pre-pandemic, but also background rates during COVID-19 circulation, but before the vaccines were introduced, to somehow address the circulation of the virus and also the role of the virus in triggering those adverse events. Does that answer your question, Jim?
Yeah, it's just how often that pre-vaccine, post-pandemic data is available rapidly. But thank you.
This seems to relate to a question also from PD Aria. I don't know whether that's the full name, but maybe Catherine, can you talk about that? Like, can you speak about the challenges of observed analysis, expected analysis for conditions that actually associated with the infection, such as anosmia and COVID-19 infection? Did you take any approach there?
That would be the case also for myocarditis and probably for some of the cardiovascular events like TTS. I don't know, Maria, did we do any adjustments in the observed to expected for infection?
So we did perform some, again, sensitivity analysis when an exploratory analysis where we used incidence rates from early pandemic. So and try to also see how that affected the ratios. But again, it was exploratory because of all the challenges of defining these rates. But yeah, it's something that we had in mind when running and interpreting the results.
Okay, thank you. Then I would like to continue with a question, another question from Rebecca Chandler. Rebecca, you are interested to see how an OE analysis was used to dismiss a signal. Did this happen by any of the agency, given, especially given the limitation of data and heterogeneity of background rates? Could you also dismiss?
Maybe dismiss is a bit strong, but I think and I'm saying again what I said earlier, which is it needs to come with complementary approaches probably. But I think you were Brenda or Narayan, you're about to speak.
Yeah, thanks. I was just going to agree with you on that. We wouldn't dismiss a potential safety signal with these, and I think we look at the observed to expected analyses as, not being able to definitively rule in or rule out a safety signal. And so I think, as you mentioned, it's kind of early in the process of evaluating the safety signal, and it's intended to provide further context for it. And so we did do another analysis, which is in the backup slides for my presentation, but we looked at a potential safety signal for increased mortality after COVID-19 vaccination. And this was in response to media reports about misinformation regarding VAERS reports of deaths after COVID-19 vaccination. So in the media, people were attributing the number of death reports to, people being killed by the COVID-19 vaccine. And we, understood that was a misinterpretation of VAERS data, and we wanted to provide more context. And so we performed observed to expected analyses comparing VAERS reporting rates for deaths to US mortality data from before the pandemic. And in our analysis, we found that the reporting rates were much lower than would be expected for all-cause mortality based on the background rate. And you can't prove an absence of a safety signal here. We're just providing more context, and so we can't rule it out, and we would still be consider potential safety signals that arise in the future. But our data were consistent with the absence of a safety signal here. So that's just one example where observed to expected analyses were negative. But again, we can't definitively rule anything out with it. Over.
Thank you. And I realize we are at the end of the hour. We do not have all questions addressed, but if people want to reach out, we can try to get an answer to them. We'll capture those questions. I would like to thank our speakers and contributors and listeners of today for the presentations and sharing their lessons learned. And thank you, and have a good evening or day.